the Faculty of Medicine, a professor at the Faculty of Medicine at the Université de Montréal. The present event is being recorded, as you just heard. So I'm a researcher in cognitive neuroscience. I'm also part of the team at the Vice Principal Office Research here at the Université de Montréal. And happy to welcome you to the annual, the 23rd year of the annual award entitled the Justine and Yves Sergent. And I will introduce the award briefly. Then we will welcome our 2022 awardee, Professor Heidi Johansen-Berg, who will share with us some of her work and contribution to the field. And then we'll have a brief ceremony, which will include the offering of the award to Professor Johansen-Berg, but also the other award, to the postdoctoral award that will be also provided. So the Justine and Yves Sergent award this year is being offered to Professor Heidi Johansen-Berg, and I will come back to the awardee. But first, let me remind you that the Justine and Yves Sergent Fund is a fund that was endowed at the Université de Montréal in 1998 by the family of Yves Sergent and Mrs. Edith Loubier. Mrs. Saint-Jean, the mother of Yves Sergent, who was also the adopted mother of Justine Sergent, passed away some years ago, but she was present for all the award events at the time when she was present. And the year after, Mrs. Edith Loubier also was deceased. Mrs. Loubier is well known in Montreal, both at the Université de Montréal, where she is an honorary member of the Faculty of Medicine, but also at McGill University, where Mrs. Loubier is honorary member of the Department of Psychiatry. And she certainly was very active in supporting research in the area of brain research, brain science. She was, in fact, Order of Canada in 1996 and Officier de l'Ordre National du Québec in 2007. And again, Mrs. Loubier met many of the awardees because she was also present each year. The fund, in fact, is to the memory of Justine and Yves Sergent, who both passed away on April 12, 1994, in the context of a long-standing, profound misunderstanding in her academic milieu, including allegation of ethical misconduct, for which an inquiry was suspended 3 years after her passing away without having raised any evidence. Justine Sergent was born in Lebanon, and when she arrived in Montreal, she was a primary school teacher. She met Yves Sergent in Lebanon. They spent just some months in France, and then both of them, Yves was also a psychologist in the school system, came to Montreal. And the career of Justine Sergent started when, at one point, when she enrolled herself at the master level, but when she finished her PhD, there was only 10 years between the moment she finished her PhD and the moment she passed away. And the whole production and contribution of Justine Sergent was in those 10 years. Justine Sergent was someone who liked music, and it showed in her research, but they were certainly very close together, Justine and Yves Sergent. They had no family here. They had no children. And so the brain science was essentially her and their whole lives. Justine started by looking at the human split brain cases, and there is a renowned article in Brain, which is a new look at the human split brain, but essentially her contribution was associated with face and object perception. She did a lot of, she did the first PET-based neuroimaging studies there. And she also looked at the distributed neural network underlying musical, sight reading, and keyboard performance. And I think if Justine Sergent has to be remembered with only one concept in cognitive neuroscience, it's the concept of hemispheric cooperation and not hemispheric specialization. And she certainly introduced this concept of the two hemispheres cooperating one with each other. At one point in the difficult context I just mentioned, this was the last writings of Justine Sergent, and they made the decision they made. So that with the family decided to transform this this situation into a celebration of women in excellence in cognitive neuroscience. So the Justine and Yves Sergent Award honors each year a woman researcher who has developed an international reputation in the field of cognitive neuroscience and brain imaging. The management committee is chaired by myself with the Vice Dean Ekat Kritikou, who will be invited to offer the award later in the event today, and Dr. Pierre Rainville, who's in the team leading the research center at the University Research Center on Aging here at Université de Montréal. The scientific committee that selects the awardee each year was composed this year of Dr. R.G. Illis from Johns Hopkins, Rita Hari from Finland at the Aalto University, and Cheryl Grady from the University of Toronto. And I accompanied this committee as well. The first Justine and Yves Sergent Award was offered in 1999, and Dr. Leslie Ungerleider, who passed away 2 years ago, was the first recipient. But among the latest recipients, Angela Frederici in 2019, Maria Luisa Corno-Tempini in 2020, Nancy Canwisher last year, who welcomed the award. And this year, 2022, it is my great pleasure to introduce you Professor Heidi Johansen-Berg, who will be talking and sharing with us some aspects about rewiring the brain, imaging and stimulating adaptive brain plasticity. So Professor Heidi Johansen-Berg is a professor of cognitive neurosciences. She's a Wellcome Trust Principal Research Fellow and Director of the Wellcome Center for Integrative Neuroimaging at the University of Oxford, England. She was born in Liverpool, so another great result, so to say, of the Liverpool to the world, or a gift of Liverpool to the world. She did her undergraduate studies in experimental psychology and philosophy at St. Edmund Hall at the University of Oxford. She did her doctorate in neuroscience at the University of Oxford. She was in her career chair of the very important and respected Organization for Human Brain Mapping. And she knows Montreal, among others, because she was a visiting researcher at McGill University, and she was also a visiting professor at Oslo University in Norway. So really, we're the scientific committee was unanimous and enthusiastic in nominating Professor Heidi Johansen-Berg as the 2020 recipient, and I will offer some minutes or 45 minutes to Professor Johansen-Berg to share with us, and then we will be back in order to remit the award. Professor Johansen-Berg, you have the next 45 minutes.
Thank you. Thank you very much. Merci beaucoup, Yves. Can you see my slides okay and hear me okay?
It's perfect.
Perfect. Wonderful. So thank you so much for this great honor to be a recipient of this really wonderful prize. I feel really honored to be associated with the work of Justine and Yves Sergent and to follow in the footsteps of such great predecessors who've been the prize in recent years. And it was lovely to hear you speak about many of those, as well as to speak about the work of Justine Sergent. So merci beaucoup to you and the committee for having given me this great honor. It's super to have an opportunity to talk to you all about my work today. I'm sorry not to be able to join you in person there in Montreal. I would have loved to do so, even though I'm sure it's quite cold over there. We even have a little bit of snow here in Oxford, which is quite unusual, but I'm sure it's colder over in Montreal. But I do have a great fondness for the city of Montreal and would love to visit next time I'm over there. But thank you for joining. I'm going to be talking to you about our work using brain imaging to understand brain plasticity, and in particular to understand rewiring in the brain. So in my group, we study brain plasticity or brain change in a number of different contexts. So how the brain changes when we learn new skills or change our lifestyle or adapt to changes in the body or the brain. You know, all of these involve brain plasticity, and we're interested to see whether by understanding basic mechanisms of brain plasticity, can we apply that knowledge to help improve clinical outcomes, for example, in the face of brain damage. And just to give you an overview of what I'll be talking about for the next 40 minutes or so, I'll start with some of our work on investigating structural plasticity, so changes in the physical properties of the brain, then talk about some of our work on functional remapping, and then go on to talk about some clinical studies where we've tried to apply that knowledge to improve motor recovery after stroke. So first of all, structural plasticity, this idea of rewiring the brain. And I was interested to see Yves illustrate some of Justine Sergent's work on split brain patients, because it was interesting reading through some of her previous work in preparation for today to see how much she'd contributed to our understanding of the hemispheric commissures and the importance, as Yves pointed out, of connections between the two hemispheres. And the model that she used to study the importance of these connections was indeed behavioral studies in split brain patients. But it was really illustrating this idea that the connections in the brain, the anatomical pathways in the brain, are really key to understanding brain function. And that's something that we've been interested in my group, and in particular to investigate whether the changes in the physical properties of the brain connections might underlie learning and plasticity. And this illustrates why that way of thinking would make sense, because if we think about the physical properties of white matter pathways in the brain, so things like the quality of the membrane or the myelin sheath around the axons, properties like the diameter or packing density of the axons, as we vary any of those physical properties of the axon bundle, that will have consequences for the physiology of that bundle. It will influence things like the speed or quality of signal transmission, as well as the brain's capacity to synchronize across a distributed neuronal network, and that in turn will impact on behavior. So one way in which the brain might change behavioral outputs is through altering the physical properties of the white matter. And that's something we've been interested in studying in my lab. Does plasticity and learning occur in part through changes in the physical property of the white matter, which can then infer functional and behavioral benefits? And we first sort of stumbled really upon this notion when my student Jan Schultz, who's illustrated here, did a study more than 10 years ago now, in which we asked the question, does the white matter in the brain change as a result of experience and learning? So in this study, we took naive, healthy adults, young healthy adults, and trained them on a new task. In this case, the new task was learning to juggle. And they were trained on this task over a 6-week period, and we scanned them before and after that training using diffusion imaging and structural MRI scans to quantify changes in brain structure in association with that training. And what we found, which is illustrated at the back here, is first of all, an increase in the volume of the grey matter, as shown by the red clusters here. And importantly, these were in areas in the medial, occipital, and parietal lobes that we know to be important in visuomotor integration, so particularly important in reaching and grasping to objects in the periphery of visual space. So we found changes in cortical grey matter in those areas, but then the important observation is the area shown in blue here, which is that in the underlying white matter, we found a change in a diffusion MRI measure of fractional anisotropy. So we found a change in the tissue microstructure of white matter fibers underlying the task-relevant areas of the cortex. So this provided at the time the first evidence that the healthy adult human brain, white matter, undergoes experience-dependent structural changes. So when we learn a new task, that not only changes the structure of the cortical areas involved in that task, but also changes the underlying white matter pathways. So this was an exciting observation, but it also raised many questions. And in particular, it raised questions about what exactly we're measuring with our non-invasive imaging techniques. And again, I was interested to see that this was a type of question, one of the types of questions that Justine Sergent was interested in her case, in the context of PET. So when she was doing many of her studies was in the early days of PET, and there was great excitement about this new functional imaging method that many people were picking up. But I found in this very interesting review article that she published a sort of ahead of its time, really, word of caution on what are we actually measuring with these imaging metrics, and are the assumptions that we're making about what we're measuring in the case of PET, brain functional surrogates, are they really justified, and should we think more carefully about what it is that we're measuring? So we've thought, similarly, in the case of our structural imaging measures, are the assumptions that we make about what we're tapping into really justified, and should we think more carefully about what it is that we're measuring with our MRI measures of white matter? So diffusion MRI, as you might know, is a technique that's very popular to use in interrogating the white matter, and it's sensitive to change. It produces a number of different parameters which can vary across groups or vary over time, but trying to understand what those non-invasive imaging metrics are actually measuring at the biological level is a real challenge. And this cartoon here just illustrates some of the ambiguity in those measures, because this illustrates a number of different scenarios which could give rise to an increase in this measure of diffusion fractional anisotropy. So fractional anisotropy, or FA, is one of the metrics that we can derive from diffusion imaging. It tells us about the directional dependence of water diffusion in this tissue, which is a useful measure, but it's a non-specific measure. So this just illustrates a number of different scenarios, all of which would give rise to an increase in diffusion FA. So for example, if there was increase in the packing density of the axons, a decrease in the diameter of the axons, increased coherence in the axonal organization, or on these scenarios over on the right here, changes in the myelin sheath around the axon, which could include both formation of new myelin on previously unmyelinated axons, or thickening of myelin on previously myelinated axons. So any one of these scenarios would give us a measurable increase in FA. And we can't differentiate those different possibilities using our non-invasive imaging metrics alone. We've been particularly interested in these possibilities on the right of myelin plasticity occurring as a result of activity, because even at the time of our juggling experiment more than 10 years ago, there was beginning to emerge more and more evidence, often at the time from in vitro studies, that the myelin around an axon was not static, but rather could respond to activity along those axons. So we were very intrigued by the possibility that this apparent activity-dependent myelination could have behavioral relevance in the context of learning. So for the last 10 years or so in my lab, we've complemented the human imaging work that we do with parallel experiments in non-human animals, in whom we can more directly test some of these biological hypotheses. So I'm going to show you some work from my colleague Cassandra Sampao-Baptista, who was a PhD student in the lab at the time and is now a lecturer up in Glasgow University in Scotland. And during her PhD, she worked with rodents, where, for example, she trained rats. Hopefully you can see that video now, the rat performing a skilled motor reaching task. So in this very well established motor learning task in rodents, the animal reach through the vertical slit in the cage to retrieve a single food pellet. And it trains on this task over about 10 days, and you see a nice learning curve as it acquires this motor skill over a period of training. And it was established that this task causes plasticity, functional plasticity in the motor cortex of the rat But we were interested to test whether this new motor learning task also changes the underlying white matter pathways. So to do that, having trained groups of animals on this task, we post-mortem scanned the brains using diffusion imaging, so to derive the exact same measures in the rodent brain that we had previously derived in the human brain. And what we found was an increase in diffusion fractional anisotropy in the white matter that you see with this red cluster here. And importantly, it's in the white matter that's directly underneath the trained motor cortex. So if you like, this is the rodent equivalent of the human juggling result that we found. But obviously the whole point of doing the experiment in rodents is that then we can follow up and perform measurements that wouldn't be possible in the humans. So for example, immunohistochemistry, where we stain post-mortem fractions, in this case for myelin. And we were able to demonstrate that in the same area of white matter, we also see increase of myelin staining. So by doing this cross-species approach, we can demonstrate that at least some of the changes that we're seeing in our human imaging measures are likely driven by this phenomenon of activity-dependent myelination. So a new plasticity mechanism, which helps to underlie learning in the brain. And we've subsequently done a number of other studies. So just to illustrate a couple. So for example, we can use other MR techniques with slightly better specificity. So for example, magnetization transfer ratio imaging, which gives us a more myelin specific measure, where we've longitudinally scanned the same animals over time and shown that the same area of white matter shows an increase in this myelin sensitive measure with learning. And we've also done studies in which we combined the MRI with gene expression analysis of relevant parts of white matter and shown increased in expression of myelin-related genes associated with the change in our MR metrics of white matter plasticity. So putting all of that together then suggests that when the adult brain learns a new skill, not only does that elicit functional plasticity and potentially cortical plasticity, but also elicits plasticity in the white matter pathways that are relevant to that task, and that at least some of that white matter plasticity appears to be associated with activity-dependent myelin changes. So this is fascinating, but raises many, many questions over the nature of this apparent new plasticity mechanism. And one of the questions that puzzled us is, well, what rules govern myelin plasticity? So if we compare it to synaptic plasticity, obviously we've known about synaptic plasticity for decades. There's decades worth of experiments that have provided us with a very good understanding of how synaptic plasticity operates, and in particular, what rules govern synaptic plasticity. And as you will all be aware, one of the primary rules that we understand well was first described many years ago by Donald Hebb, and can be summarized as this idea that cells that fire together wire together. So synaptic plasticity, we know, cares about what we call coincidence detection. So it's a spike timing dependent plasticity. Selectively, synapses strengthen when impulses arrive close in time to each other. So one question that we had was, well, what about this myelin plasticity? Does myelin plasticity care about coincidence detection? Do active axons simply get more strongly myelinated regardless of the downstream consequences of that activity? Or do axons get selectively myelinated only if the impulses that are passing down them have some consequence downstream? So that's kind of a fundamental unknown about this kind of plasticity, and raises all sorts of interesting questions about what signals might help to allow for that type of coincidence detection to occur in oligodendrocytes, the myelin forming cells in the brain. But as one first step to start to address this question, we carried out a study exploring this question in humans in this case. And this is work from Alberto Mazzari, who was a PhD student with me at the time. And Alberto used non-invasive brain stimulation as well as imaging in human volunteers to test this hypothesis. So the way he did this experiment was to use a paired pulse TMS protocol, which is illustrated schematically here, where we take pairs of pulses, and we have pairs of pulses over the ventral, with one pulse over the ventral premotor cortex in one hemisphere, and a second pulse over the primary motor cortex in the other hemisphere. And this is a well-established, well-described phenomenon where we can stimulate these two areas, and we can adjust the timing of between those two pulses. So in a Hebbian protocol, shown here in green, where those pulses are in quick succession with one another, we know that that type of stimulation pattern physiologically strengthens the pathway between the ventral premotor cortex and the primary motor cortex. Versus in yellow, we can put the same pulses into the system. We can apply the same pulses, but separate them in time into this controlled non-Hebbian stimulation pattern. And we know from previous studies that that protocol doesn't physiologically strengthen the pathway. So the question is, does myelin plasticity care about whether the stimulation is Hebbian or not? So to address this question, Alberto combined these types of paired pulse TMS protocols with a brain imaging readout, which was sensitive to myelin, in this case, magnetization transfer myelin mapping. So by scanning people before and after either a Hebbian or a non-Hebbian stimulation protocol, we could ask the question, does the physiological plasticity induced by the TMS protocol influence the degree of myelin plasticity that is detected? And the result that we found is shown down here on the bottom, where the areas in red in the white matter, we find a correlation between the change in our myelin metric and the degree of physiological plasticity evoked by the stimulation protocol, but specifically in the Hebbian condition. So putting all of that together, it suggests that specifically Hebbian stimulation induces myelin plasticity, and the greater the amount of functional plasticity produced by the stimulation, the greater the amount of myelin plasticity, whereas we don't see that relationship with the non-Hebbian stimulation, which doesn't produce functional plasticity. So that suggests then that the myelin plasticity does indeed seem to care about the same kind of rules that govern synaptic plasticity. So could indeed potentially function as a useful adaptive plasticity mechanism that's relevant to learning. So just summarizing this first third of the talk then, we've shown that experience shapes white matter microstructure in the human brain, but relating these gross structural changes to the underlying biology is challenging. And our animal studies suggest that white matter plasticity may relate in part to activity-dependent myelination, but this is a very new and emerging plasticity mechanism that we understand very little about, but we're beginning to try to probe the rules that govern myelin plasticity, and recent studies suggests that myelin plasticity might indeed follow Hebbian rules. So coming on into the second part of the talk, where I want to tell you about some of our work on functional plasticity using functional imaging, and in particular how we've used functional imaging to look at plasticity of the sensory cortex. And this is work from James Kolosinski, from when he was a PhD student in the lab. And this is when this was a study that we did, one of the first studies we did when we first got 7 Tesla MRI scanner, where like many people, we were looking for ways to exploit high field MRI. And one of the things that high field MRI is especially good for is high spatial resolution functional MRI mapping. So to exploit this capability, we looked to see whether we could map individual digit representations in the sensory cortex, and that's what's illustrated here. So James optimized a very nice protocol in which participants would sequentially move the fingers of the hand in a so-called phase encoding paradigm, where they just repeat the movements across the 4 fingers repetitively over time. And what that elicits is a traveling wave of activity moving up the sensory cortex, like you see in the movie there. So then we can simply categorize voxels along the sensory cortex based on their preferred finger. And when we do that, we see this very beautiful individual digit maps along the sensory cortex, which display this very characteristic topology with the 2nd, 3rd, 4th, and 5th digits represented as you move up the sensory strip. And we were interested to explore how these maps varied across people and across time. And that's illustrated here in just 3 participants. There's more participants in the paper if you're interested to look. And what we found was that was a few interesting things. So one is that basic topography going from bottom to top is pretty well preserved across participants, but the precise size and shape of the digit maps varies between participants. But within participants over time, so this is the same people scanned a day later or 4 weeks later, we get a pretty good preservation of the specifics of the digit maps over time within a given participant. So to summarize, the maps are variable between subjects, but are highly reproducible within subjects. So what this means then is that this is quite a useful paradigm with which to ask the question, under what circumstances could we change those digit maps? Given they're pretty stable over time in general within people, can we shift those digit maps if we alter people's experience over time? So to ask that question, so to assess the short-term plasticity of digit maps, James carried out a study in which people came into the lab for multiple different scans, and then in various 24-hour periods, he compared on the left a control 24-hour period where subjects went about their business as usual, to on the right a gluing 24-hour period where he glued together the index and middle finger of the subject's hand using a special surgical glue, which could fuse the fingers together but be easily removed the following day, but would leave the skin surfaces exposed as normal. 24 hours of fusing those two digits together, and then we could look at how whether there were changes in those digit maps associated with this gluing protocol, which would have altered their experience during that 24-hour period, and obviously counterbalance the order of these different conditions. So for those of you who are familiar with the sort of classical neuroscience literature from non-human primates, this might sound like a familiar type of experiment, because there were many studies from some time ago where researchers would suture together the digits in primates and then electrophysiologically map their sensory representations over months of altered behavior. And based on that literature, we had expected that when we stuck together digits 2 and 3, we would see a corresponding coming together of the cortical maps of digits 2 and 3. So this is illustrating a schematic of the cortical maps of the 4 digits along the central sulcus. And what we expected was that as we glued together digits 2 and 3, we'd get an increase in the overlap between the cortical maps of digits 2 and 3. But interestingly, that's not what we found. What we found was it was a change, but a pattern of change. So after just 24 hours of gluing the digits together, we quantified the amount of overlap between the cortical digit maps of each of the pairs of digits. And what we found is illustrated here is that for the 2 control conditions, there was no change as we'd expected. These maps are stable over time. But for the scan taken post-gluing, there was a dramatic change in the degree of overlap of the digit maps. But interestingly, the specific pattern of the change was that we didn't see a change in the overlap between the 2 digits that were glued together, but what was changing was the other pairs of digits. So we saw less overlap between 3 and 4 and more overlap between 4 and 5. So going back to our schematic representation, what we found was actually this. So after we glue together 2 and 3, 3 and 4 get further apart and 4 and 5 get closer together. So this was very intriguing and suggests that something a bit different might be happening with a short-term change in experience, such as we're manipulating here. But because this was a surprising result, we wanted to follow up to see if we could make sense of it by testing the behavioral consequences of this remapping. So we made specific behavioral predictions based on the neural predictions that we'd observed. What we predicted was that given the change that we had seen in the cortical maps, we would expect that the digit pairs where we had observed increased cortical overlap, 4 and 5, subjects should get worse at discriminating sensory inputs to those 2 digits. Whereas for the digit pair 3 and 4, where we'd seen a greater separation of the cortical maps, subjects should get better at discriminating sensory inputs to those digits. So James then ran a new experiment to test those behavioral predictions using the same gluing manipulation. And what we found was that the behavioral change did indeed fit with the predictions from the neural change. So the task that he used was a temporal judgment task where you deliver pairs of tactile stimuli to pairs of digits, and the subject has to report which digit was stimulated first. You can generate a nice psychophysical curve and quantify how good each subject is at discriminating tactile inputs to those 2 digits. And what we found, so now we're plotting for the same 3 conditions, the behavioral data, so how good are you at discriminating tactile inputs to the digits? Again, no change in the control conditions, but a change in behavior after the gluing condition. And specifically, For the pair where we got less cortical overlap, they got better at discriminating. And for the pair 4 and 5, where we got more cortical overlap, they got worse at discriminating. So this short-term change in sensory motor experience evokes rapid remapping of the overlap between cortical digit maps that has consequences for behavior. So this was an interesting result, but it was also particularly intriguing given that in other work, we've shown that the basic topography of those cortical digit maps is remarkably resilient to altered experience over long timescales. And for this second point, I'm going to turn to some work that I conducted with my colleague Tamar Macon, who was a postdoctoral fellow in the lab and is now a professor at Cambridge University. And Tamar brought to the lab an interest in plasticity following limb amputation. And you're probably familiar with the sort of prevailing view of what happens in the brain after limb amputation, which very much focuses on the consequences of sensory deprivation. So this is a sort of Penfield's homunculus that will be very familiar to you all in Montreal, no doubt. And as you will be aware, when we remove the sensory input, say, of the upper limb, there is evidence that you get sort of invasion of neighboring body part representations into the vacated sensory motor cortex. And so this was really the prevailing view, was that when you remove the input to an area of sensory motor cortex, the neighboring body parts invade that area. But what we did with Tamar was look at the problem in a slightly different way to try to understand what is the representation of the missing limb. And as you will be aware, many people who experience limb amputation continue to perceive a phantom limb. So a very vivid, often a limb that they can control the movements of. So what Tamar did was take people who experience phantom limb post-amputation and ask them to move that phantom limb in the fMRI scanner so she could look at the representation of the phantom itself. And what she found, interestingly, was actually a very well preserved representation of the phantom hand. Obviously, the hand is not there, but these individuals can move their hand voluntarily. And very importantly, asking them to move their hand is not the same as asking them to imagine moving the hand. Neuronally, you see a very different pattern of activity. But what you're seeing here is the degree of activity for two-handed controls moving their non-dominant hand, and the activity for amputees moving their phantom hand. And what you see in the graph is the quantification of those different activity patterns in a non-biased region of interest. And you can see that there's no difference between the degree of activity between amputees and two-handed controls. Whereas, interestingly, if you take this one-handed control group here, are people who were born with only one limb, so-called congenital amputees, who don't typically experience phantom limb. If we ask them to move their missing limb in the scanner, they think it's a crazy question, but if you ask them to try to move the missing limb in the scanner, you can see that you don't see that same degree of representation. So in amputees who experience a phantom limb, the representation of that phantom is just as strong as the representation of an intact limb. Interestingly, in that paper, we explore the relationship with phantom pain, but I'm not going to go into that today. But if you're interested, I'd be happy to tell you more about the fact that this also seems to be preserved by the experience of ongoing phantom pain. But the question I'm going to follow up here is, well, okay, appears to be a preserved representation of the phantom limb, but to what extent is that representation organized? And to do that, we're going to use this individual digit map technique that I described to you earlier. So as you saw in the previous study, we can use high field MRI to map representations of individual digits in the sensory strip. So a study from Sane Kickert, who was a PhD student in the lab at the time, who's now a postdoc in Zurich. Sane used the same digit mapping technique to look at individual digit representations in controls and in a case study of a few upper limb amputees. It was quite hard to find upper limb amputees who were safe to go in the 7 tesseract MRI scanner, because many of them have experienced traumatic accidents to experience their amputation and have various implants, etc. But also we wanted to find individuals who could voluntarily move individual digits of their phantom limb. So this is data from a couple of the control subjects, just to illustrate what the activity patterns look like. So again, we're using color coding to represent in this case, 5 different digits. So in this experiment all 5 digits are moved. But you see that same topography that we saw in previous experiments with digits 1, 2, 3, 4, 5 organized along the sensory strip. But then interestingly, so that's a couple of example control participants. What we found in amputees was that despite the fact that these limbs had been missing for 20 or 30 years, we saw just as much evidence of topography representations in amputees. So when the amputees were asked to move the individual digits in the fMRI scanner voluntarily, we saw preserved digit representation. That's shown by this here, and in the paper there's various ways of quantifying the organization of those maps and demonstrating that they're just as organized in the amputees, despite the fact that the fingers themselves have been absent for many, many years. So in summary then, short-term changes in sensorimotor experience can rapidly alter cortical maps, but that basic topographic organization seems remarkably resilient to altered experience, even over long timescales. So for the final part of the talk, I want to briefly tell you about some of our work, more clinically oriented work, looking at whether we can stimulate plasticity. And here our particular area of interest has been trying to improve movement recovery after stroke. And one approach that we've taken is to use non-invasive brain stimulation to enhance learning or rehabilitation. And here I've collaborated for many years with my colleague, Charlie Stagg, who's modeling the brain stimulator in the picture here. And what Charlie showed through her PhD and postdoc work is that we could use facilitatory brain stimulation to enhance learning or rehabilitation. So the logic here is that rehabilitative training, just like learning, relies on processes of brain plasticity. So if we can find interventions of plasticity, we can then test whether those improve rehabilitation. And one intervention that we've been interested in is anodal transcranial direct current stimulation, or TDCS, which is a technique that's being illustrated in the photo here. And what Charlie has shown is that when we apply anodal TDCS to the primary motor cortex, it facilitates plasticity and learning in healthy people. And we've done this through a number of different types of demonstrations. So Charlie's shown, for example, using spectroscopy, that this kind of brain stimulation reduces GABA in the motor cortex, and that's important because GABA is the primary inhibitory neurotransmitter, and we know that removal of inhibition is one mechanism to facilitate plasticity. We've shown using fMRI that this type of stimulation increases the activity of the stimulated M1, and behaviorally in healthy subjects it speeds up motor learning for short timescales. So putting all of this together, there's a good case for testing whether in patients post-stroke with movement difficulties, could you combine rehabilitative training with this kind of brain stimulation in order to boost plasticity and thereby improve rehabilitation outcomes? So one way in which, so we could, for example, try to use this kind of anodal TDCS to try to balance the brain post-stroke. And this was first suggested many years ago. This is a study from Leo Cohen's lab at NIH from some years ago, a series of case studies of patients post-stroke, where they showed that applying this type of brain stimulation for a single session consistently speeds up movement times in patients post-stroke. And by combining the stimulation with functional imaging, we were able to help explain why these benefits happen, showing, for example, that you see increased functional activity in association with movement during stimulation, and that correlates with behavioral gain. So the greater the increase in activity evoked by brain stimulation, the greater the behavioral improvements that you can observe with stimulation. So with all of this in mind, then we designed a longer-term randomized controlled trial in chronic stroke patients, where people were given 2 weeks of motor training, which was paired with either real or sham brain stimulation in a double-blinded fashion. And what we found was that the addition of brain stimulation to rehabilitation boosted outcomes. So the stimulation group is shown here in green, and a control sham stimulation group who received the same training in blue. And you can see that all the way out to a 3-month follow-up time point, you see greater benefits in a clinical score in the group that received the rehabilitation while having their brain stimulated in this way. And we showed using imaging that this was associated with greater functional recruitment of the premotor cortex in the stroke hemisphere. So this suggesting that there is some promise in the prospect of using brain stimulation to boost plasticity mechanisms and improve rehabilitation outcomes. More recently, we've been interested into whether we could, instead of using extrinsic brain stimulation to modulate the brain in this way, could we tap into the brain's intrinsic capacity for activity modulation using neurofeedback? So the idea behind the neurofeedback approach is illustrated schematically here. So you'd have a participant, for example, in the fMRI scanner, performing a particular task. You're reading out their brain activity signals in real time, analyzing them very quickly, automatically on a computer, and then feeding back a visual representation of those brain activity signals to the subject, where they're instructed to modulate that visual display in some way, which corresponds to modulating their brain activity in a particular way. So in a very simple-minded way, then we could think, okay, could we use this type of feedback to train people to make their motor cortical activity more lateralized, given that that's what seems to benefit patients post-stroke. So following a stroke, patients tend to show bilateral patterns of activity. We know that more lateralized activity is associated with better outcomes. So can we normalize or rebalance those activity patterns through the use of neurofeedback? So to do this, my colleagues Heather Niedli and Yunying Huang carried out an fMRI neurofeedback study in which people were told to move one hand, and they were displayed in real time moving bars, where the red bar represents the activity in the contralateral motor cortex and the blue bar represents the amount of activity in the ipsilateral motor cortex. And they're simply told to try to move and while maximizing the height of the red bar and minimizing the height of the blue bar. And it turns out that both healthy older adults and chronic stroke patients can indeed use this kind of feedback in real time, push their activity to a more lateralized pattern. So this is an interesting proof of concept, but in these studies, which just look at a single session of feedback, as soon as you take the feedback away, the activity reverts back to normal. So obviously, for this to have any clinical benefit or clinical relevance, we want to see whether we can train patients to produce more lateralized brain activity patterns, and then that to persist even in the absence of feedback. And that's what we've tried to do in a longer term study in chronic stroke patients, to see whether we could see cumulative effects of repeated sessions of neurofeedback over time. So this was, again, a double-blind randomized control trial that involved many different colleagues in chronic stroke patients. And patients came in for 3 days of fMRI neurofeedback, and they were scanned before and after the feedback, and they had various other assessments, including clinical assessments at all of those time points, as well as at a 1-month follow-up time point. And what we found, first of all, so to ask what's the effect of the neurofeedback, how does the neurofeedback shift their brain activity, and do we see cumulative effects of multiple sessions of neurofeedback? So the fMRI results during neurofeedback are illustrated here. So what we're plotting here is that laterality index, so quantifying the laterality of functional activity between the two hemispheres for the real neurofeedback condition in green and the sham neurofeedback condition in gray. And so we have 3 days of testing for each of our conditions, and we have 3 blocks of feedback on each of those days. What you can see in the real neurofeedback condition is that within each testing session, subjects do indeed improve, so they can make their brain activity more and more lateralized within a testing session, but there's no evidence for cumulative effects over time. So patients can use neurofeedback to increase the laterality of their functional activity. And so we see increased laterality during neurofeedback, but there's no evidence for cumulative effects over days. So they go back to the same position on the next day each time. So we see short-term effects of neurofeedback. Interestingly, though, as with all neurofeedback studies, there's a lot of variability in how well people can do neurofeedback. It seems some people are quite good at neurofeedback and some people aren't. And what we find is that in patients who are good at using the neurofeedback to control their brain activity while the neurofeedback is there, we see transfer of those changes even when we take the neurofeedback away. Whereas in the people who weren't very good at using the neurofeedback while it was there, we don't see any persistence of the changes in laterality after we take the feedback away. So as is often the case with these neurofeedback studies, there seems to be a lot of individual differences, a lot of variation between people. And when it works, there's a hint that longer term changes in brain activity might persist. But obviously the key challenge in this is to know when it will work and when it won't work. But there is a hint that those who respond to neurofeedback show transfer effects. So what about longer term effects then when we look at the changes in behavior over time? Well, in this clinical trial, we had a primary clinical outcome which didn't shift at all with the neurofeedback. So we saw no evidence of clinical gain as a result of neurofeedback. But there was an interesting hint of improvement in specific in subcomponents of the. So this is the Jebsen-Taylor task, which is one of the timed clinical tasks that we used, where we did see for that gross component of the Jebsen-Taylor task an improvement in the real neurofeedback group in green compared to the sham group in grey. So again, no dramatic effect, no effect on our overall clinical measure, but a hint that with certain types of movements, we are seeing an improvement, albeit in the patients in whom the feedback works. And what about the brain imaging results? Do we see any evidence that this type of training alters the brain? Well, intriguingly, we do see a clear effect on white matter microstructure. So here we're quantifying the asymmetry of fractional anisotropy across the two corticospinal tracts. And we see a very clear interaction here, such that the group who are receiving the real neurofeedback, their corticospinal tract white matter microstructure becomes more symmetrical as a consequence of the neurofeedback. And that's driven by an increase in FA in the affected hemisphere, as we would expect. And interestingly, this effect correlates quite closely with the neurofeedback response. So again, in those individuals who can use the neurofeedback effectively, we're seeing this brain plasticity response. So putting all of that together then for the clinical part of the talk, we see evidence for beneficial effects of brain stimulation, TDCS, as an adjunct to rehabilitative training, and using neurofeedback to shift activity patterns. We've shown that neurofeedback can indeed shift movement-related activity towards a more lateralized pattern, but as yet there's no evidence that this consistently persists after feedback is removed, albeit a hint as in those who could use the feedback most effectively. It therefore doesn't result in clinical gains despite this clear evidence for changes in white matter microstructure. So in summary then, experience shapes the structure and function of the human brain, and relating imaging changes to underlying biological events can be challenging, but is an important thing to grapple with. And we hope that by understanding brain plasticity, we can help in the design of new rehabilitation treatments. I'd finally like to thank all of my colleagues whose work I've had the pleasure of sharing with you today, and thank my funders, and once again, very much thank Yves and colleagues for the wonderful recognition of this prize. Thank you very much.
Merci beaucoup, Professor Johansen-Berg. This was really a great and outstanding demonstration of the contribution to a better understanding of the basic mechanisms under neuroplasticity, and in fact it applied to motor and sensory aspects, as you mentioned, but are probably expandable to the rest of cognitive processing into the brain, both as a reaction to some events in life or as we move forward in life, since aging is a everyday brain plasticity challenge, in fact. So merci beaucoup, and certainly demonstrates that you and your team, congratulations, certainly deserved the Justine and Yves Sergent Award that the scientific committee declared this week. And in this sense, we will not have this event is not accompanied by a question period. We will ask people maybe to get in touch with you. I don't want your email to be too much overwhelmed, but still, since either in a next meeting or whatever. But we will immediately go to the very brief official part of it, which is the official remittance of the award. And I will ask Dr. Ekat Kritikou, who's Vice Dean Research at the Faculty of Medicine of the Université de Montréal, where the fund is in fact managed, to say some words and to present to you virtually the certificate. Dr. Kritikou, Ekat.
Thank you very much, Yves. I'm really delighted to be with you today. It's my second year as Vice Dean that I present this award, and I learned so much. I find the brain so amazing, although not a neuroscientist myself, but this award is also an opportunity for me to understand what you're doing and what our researchers are interested in. So I'm very happy to be with you today to celebrate this award. I just wanted to reiterate this award is really a celebration, as Yves mentioned earlier on, a celebration to really to the memory and the spirit of Justine and Yves Sergent. And I'm very happy today that we have with us Dr. Johansen-Berg for this award. I think your talk really highlights that we want to give to this award to really outstanding researchers in the field of cognitive neurosciences. And congratulations, I think you have really shown all the unknowns, or some of the unknowns we don't know about our brain, but also going from the very fundamental to clinical neuroscience. I think it's an important demonstration of what we will be able to do in the future on manipulating the brain and having solutions for different diseases. So I'm delighted to present this award to you, and I will invite you to open the envelope and show the award to our audience.
Wonderful. Thank you so much.
In fact, the problem is that there is a strike in UK relative to mail delivery, and I think the award you'll have to look at it, Professor Johansen-Berg, first, and you will receive it very, very soon. But it was posted 3 weeks ago, but unfortunately the conditions did not allow it to arrive. So it will come soon. And thank you so much, Dr. Kritikou, for having presented the coming certificate, which of course comes with the $10,000 award, Canadian dollars, which is remitted to the awardee each year. So again, congratulations, Dr. Johansen-Berg, and thank you, Dr. Kritikou. And now, if you don't mind, I will take just 2 minutes to present the appended award, which is the postdoctoral award, Justine Sergent. This is a new award since last year. And the postdoctoral award is a partnership with the Fonds de Recherche en Santé du Québec, FRQS, and the Fonds de Recherche Québec Santé, en fait. And it is offered to one of the postdoctoral awardee from the FRQS, who is working in the area of cognitive neuroscience. And there is a scientific committee, and I would like to thank Professor Shirley Fecteau from Laval University and Professor Anaïs Ansaldo from Université de Montréal for having doctoral fellows of the Fonds Recherche Québec Santé, the one that will be receiving, and Dr. Colleen Hughes will be receiving the award. It so happens that Dr. Yu is with me in my office. I will make a little bit of, just a minute. I will just remove my background here, and I would like to present you Dr. Colleen Hughes. And Dr. Hughes being here, I will remit to Dr. Hughes the certificate for the postdoctoral, including the award that comes, which is an extra addition to her postdoctoral fellowship that you can use to travel or to whatever you wish for.
Okay. Thank you.
Merci beaucoup. So on this, it's you know, making these things virtual is a little bit complicated. But again, thank you so much, Professor Johansen-Berg, Heidi, and you've been visiting professor to Montreal, to McGill, and I'm sure that many people in Montreal will be happy to welcome you in any future travel to Montreal. So again, thanks to all of you. It's 1 o'clock. We wanted to keep that on time. And thanks again to everyone for having participated in this event. Merci beaucoup, Dr. Critico. Thank you.